Welcome back, students. This is Module 2.3.3, Cellular Respiration, Glycolysis and Fermentation. We're going to start by sharing with you some definitions of cellular respiration. The first one is a long definition to understand, but not necessarily to memorize. So cellular respiration may be defined as the enzymatically controlled, stepwise oxidation of nutrient molecules in the cell that catabolically releases and transfers the energy of the food molecule's chemical bonds to the useful cellular form of ATP. In other words, we're doing a controlled burn of food to release the energy and transform it into ATP. So the short definition to both understand and to memorize is cellular respiration is transforming energy in food into ATP so it can perform useful work at the cellular level. This process in eukaryotic cells occurs in organelles known as mitochondria. The mitochondria are considered to be the site of cellular respiration, of aerobic respiration. And in high school or other places, you may have heard that the mitochondria may be referred to as the powerhouse of the cell because those chemical reactions of aerobic cellular respiration occur within it. Now, what's important to recognize in both the powerhouse and in the mitochondrion is energy is not being produced per se. We're transforming energy from one form into another in the mitochondria. And that's the same thing that happens perhaps in a powerhouse, where you might have hydropower being transformed into electricity, or in a power plant where we're burning coal and transforming it into electricity. The energy isn't produced from scratch, it's transformed from another source. And that's exactly what happens inside the mitochondria. The mitochondria are oblong in shape, and they have a double membrane. And you should recall that this is because it is believed that they originated by endosymbiosis, though that doesn't have any great pertinence here. So you should know this basic structure, the one on the bottom. This is a diagram, a transmission electron micrograph on the top, and an interpretive drawing on the bottom. And so there's an outer membrane and an inner membrane, and then there is an inner space that's called the matrix. That's the one that looks kind of purplish in the bottom diagram. And then there's an additional space out here. I guess we could call it the outer space, very groovy. But the folds of the inner membrane are called cristae. And these folds serve to provide much more surface area where enzymes can be embedded. Therefore, in a mitochondrion of the same size with this convoluted cristae, these folds of the cristae, you can produce a lot more ATP. And as we learned previously, we need millions and millions of ATPs per second for our cells to survive. So this is a very important aspect of the mitochondria. So you should be able to be familiar with all of these structures here. To introduce how food is used at the cellular level, we're going to study carbohydrate catabolism, which is to say we're going to look at how a simple sugar, glucose, is broken down and transformed into ATP. Now, glucose of course, is very important. You learned in biochemistry it serves as a monomer for many polymers. It is also the body's preferred energy source. Your brain, for example, runs on glucose. The catabolism of other food types, remember, that aren't carbohydrates, proteins, lipids, nucleic acids, they have other metabolic pathways that merge with those of carbohydrate metabolism. We're not going to directly study those. But in an upper division biology course, you might learn more about that. But just here in this introductory survey, we'll just take it on faith that there are these pathways, and we'll learn about the carbohydrate pathway in more detail. Now, this next slide is going to show you a kind of an overview diagram. And notice it says right here, draw this. So you should draw this. Understand, though, first where you are. the blue represents the cytoplasm of the cell. This purplish sphere is the nucleus, and it has no direct role in the processes. It just serves as a landmark to show us that we are in the cytoplasm. So start up at the top, and glucose is our input, and then we have an arrow that goes into the box. The box represents a process. The process is glycolysis. And notice the outputs from glycolysis from this process are some energy, that's what this little sunburst represents, and then pyruvate comes out. And pyruvate is also in the cytoplasm. The fate of pyruvate depends upon the oxygen environment in the cell. And so we can see a fork in the road here. If there is no oxygen, then the pyruvate will undergo the process of fermentation, which also occurs in the cytoplasm. And then we can see the various outputs. You should write these down in your diagram, and we'll be talking about them in detail later. The other branch goes to this tan structure with two membranes. This represents the mitochondrion, and the red dot doesn't represent a structure, it represents a cycle that occurs in there. And then you can see the output there is this sunburst here. So we will later, after we go through this overview, apply these names. And so these names, although not very useful now, you look at them and they just look like alphabet soup. Go through the explanation of this diagram and the processes on the following slides, then come back and look at this, and this will serve as a very nice summary slide and will help to cement your understanding of the process together. So we're going to start with glucose in the cytoplasm, and it undergoes the process of glycolysis. Glycolysis, if we look at the etymology, it is literally sugar splitting. Glyco is sugar and lysis is splitting. We've seen lysis before, for example, in a hydrolysis reaction, splitting with water, how biological macromolecules, how their polymers are split into monomers. But so lysis is splitting, glycolysis sugar splitting. And remember, we're starting with glucose, which is a 6-carbon sugar. The structure of glucose is C6H12O6. The process of glycolysis is a universal precursor. When we say something like universal precursor, we mean it precedes all carbohydrate catabolism, and virtually all organisms have a glycolysis process. And glycolysis may be defined, I suppose, as the process by which glucose is split into two 3-carbon pyruvate molecules. So the 6-carbon glucose is split into two 3-carbon pyruvate molecules. And this occurs in the cytoplasm. There are enzymes there for glycolysis, and that's why it occurs there, and that's also where the glucose is. And what happens next to these two 3-carbon pyruvate molecules depends. It depends upon the oxygen environment in the cell. I should also note that the process of glycolysis produces 2 ATPs. It in fact produces more than that, but some of them are consumed in the process. So there's a net production of 2 ATPs. So 2 ATPs come out of the process. And again, as I said, what happens to these pyruvates depends on the oxygen environment in the cell. And there are two possibilities. The oxygen environment can be anaerobic or aerobic. Anaerobic means lacking oxygen. Aerobic means oxygen is present. And you might be thinking, well, I've heard of aerobic before, and I thought aerobics was a funny exercise done in a leotard. Well, it turns out that aerobic and anaerobic are also two types of exercise. And aerobic exercise is exercise that is performed at a measured, limited rate. That's why during an aerobics class, if you have a good instructor, they'll have you taking your pulse from time to time to keep it from getting too high. In other words, you want to exercise at a rate that does not exceed the ability of your body to deliver oxygen to your cells. And so that by exercising at that limited rate, it maintains an aerobic environment in the body cells. Now, anaerobic exercise, by contrast, involves a more extreme effort. For example, think of running wind sprinting, weightlifting, this extreme exercise exceeds the ability of the body to provide its cells with oxygen, and therefore the environment inside the cells is anaerobic. And so this is a way to help you to understand both exercise and to understand a little bit more about this process that we are studying now. So just remember that aerobic means oxygen is present, anaerobic, oxygen is lacking. And I know that many people are curious, so we'll talk a little bit more about exercise. And athletic training increases the efficiency of oxygen delivery to cells. One of the major mechanisms, certainly not the only one, that's improved by training is angiogenesis. Angiogenesis is the formation of blood vessels. So if you have a greater circulatory capability, the ability for more blood to reach your cells more quickly, then it's less likely that they would run out of oxygen and produce an anaerobic environment. When an organism, an animal or a human, exceeds their aerobic scope, the ability of the body to deliver oxygen to the cells, then an anaerobic environment is produced, and they'll go into something that is called oxygen debt. And that's simply when that rigorous exercise exceeds the ability of the body to deliver oxygen to its cells, requiring the use of anaerobic pathways. Boy, that was a tough one. This oxygen debt mandates that there is a recovery period afterwards. Think of a cheetah after it sprints and captures a gazelle, or think of sprinters after a long sprint, more than even 100 meters. The runners have to sit there and take a blow, really breathe deeply. But think of a 200 or a 400 meter sprint. They really do. And so a cheetah, even after it captures its prey, it can't start eating. It has to sit there and pant to fulfill this oxygen debt, even though as it sits there all the time, it is at risk of having a larger cat like a lion come and chase it off its hard-won meal, its prey that it had to capture. So anaerobic exercise is not fun. The long sprints, the 400-meter sprint, the 800-meter sprint, these are very difficult races. And before we end off on this topic, we want to show you what happens when there is an anaerobic environment in the cell. So glycolysis has occurred, pyruvate has been produced, and the oxygen is anaerobic. Then what will follow is the process of fermentation. So you follow the fork to the right in your little diagram to that rectangle, which is a process of fermentation. And here's fermentation. So fermentation occurs if no oxygen is present. The environment in the cell is anaerobic. Together with glycolysis as a precursor, those two together make up the anaerobic pathway. So glycolysis and fermentation are the anaerobic pathway. Like glycolysis, fermentation also occurs in the cytoplasm. And the waste products depends on what organism this process is occurring in. In some fungi, alcohol is produced as a waste product, alcohol known as ethanol. And in vertebrates, animals with backbones, the molecule lactic acid is produced as a waste product from fermentation. So those are waste products that come out of the process of fermentation. There is no additional ATP, no additional energy is produced in fermentation, but fermentation is required because it allows certain molecules to be reset. We say that recharged, and this will allow glycolysis to occur again. Without fermentation, glycolysis could not occur again, and death would be very rapid. But fermentation allows a limited number of ATP from glycolysis to be produced. And so that's an important outcome, because the alternative is death, and death is not good. So the net energy gain is 2 ATPs per molecule of glucose for both glycolysis and fermentation for this anaerobic pathway. So the anaerobic pathway produces 2 ATPs per glucose. And although anaerobic respiration is very inefficient, it is better than the alternative. Can't be used very long by creatures that are large-bodied aerobic creatures, such as humans and other vertebrate animals. This shows two types of fermentation. Alcohol fermentation that's produced by some fungi such as yeast and a few other bacteria. Bacteria have a very wide range of metabolic pathways, and we don't certainly even begin to address all of them that are possible. But notice the 6-carbon glucose comes in, undergoes glycolysis, produces pyruvic acid, and then it produces this waste product and also carbon dioxide, and it produces ethyl alcohol, which is ethanol. Interestingly, that is the same alcohol that is in beer and wine. This is booze. And so yeast, this is brewer's yeast that will do this and produce this alcohol. And it's very noteworthy that it is, in fact, a toxic waste product that is being produced. If we look at a second pathway in a different lineage of animals, it still occurs in the same place in the cytoplasm after glycolysis. And pyruvate are, by the way, pyruvic acid and pyruvate are being used interchangeably as synonyms here, and they are very close. Pyruvic acid can donate a hydrogen ion, and it becomes pyruvate. But anyway, we can see glucose here undergoes glycolysis, produces pyruvic acid, and then produces lactic acid. And the real critical thing that's happening right here is a little energy carrier. We're not making you learn NAD and NADH, but that's what's happening, is this is being reset so that glycolysis could occur again. And notice it's happening in both examples of fermentation here. And so that is the common process that's occurring. I do want you to really think about that. If you enjoy alcoholic beverages, that this is alcohol is essentially mushroom pee. That's overstating it, because yeast is not really a mushroom, but it is fungal waste. Kind of interesting. And just before we close this module, I want to address something called the white powder rule. And that is, some biologists will tell you that it's okay, that all societies have had some kind of way of altering reality, some kind of alcoholic beverage or drug that they take or something. And what the white powder rule is simply the expression of a belief. It's not. This is not science by any stretch of the imagination, but it is an idea that less processed stimulants and drugs are better than more processed ones. For example, the white powder rule would say that beer and wine are okay to drink, but that you should never drink distilled liquor, the hard stuff, where it's much more refined. And the white powder rule, of course, comes from the cocoa plant, where the leaves are chewed in South America as a stimulant, but if you take those leaves and process them, you can make cocaine, which is, of course, a white powder, which is very powerful. Now, the white powder rule has some problems because some of the most the implication is that nature is benign and that humans make things more dangerous. Of course, that's just silly, because some of the most poisonous and most deadly substances in the world are produced directly by nature. But nevertheless, it's an interesting idea, and just another one that's floating around out there for you to think about. That will close this module. Thank you very much.